Hello, Assalamu alaikum. In this video, we will understand about the progeria, the progeroid syndrome. And the progeroid syndrome is very rare, and in this way, the also known as Hutchinson-Gilford syndrome. So we will discuss all about this progeria, the detail. So Hutchinson-Gilford progeroid syndrome, HGPS, it is known as for short, and this is due to the single gene mutation lamina or lamin A. So this is the genetic mutation, and in this way, basically the lamina makes protein for holding the nucleus of the cell together. So they typically live until their mid to late teens or early 20s. In this way, basically important thing you should need to understand, very less lifespan as compared to the normal person, and the person will look like more and more aged due to the activation of the one gene lamin A. So lamin A gene, we will understand all these things. So let's understand about the phenotypes of progeria first, and then we will dive into the main concept to understand how it is possible. This is the person which that is the progeria containing person, the progeria syndrome. It can be alopecia, the loss of hairs, and lack of eyebrow, eyelashes, and a premature heart, and also diseases like cardiovascular diseases. On the other hand, the cerebral chin, which that is a major sign, the narrow chest, and the swollen and stiff joints. On the other hand, the age-related spots and the dry and thin skin will be appear like a aged person. The large head and the circumference of the head will be large, and small jaw and crowded teeth. That is the sign and symptom in the early age. 10 year and 10 to 20 year is the end. So in this way, here is the lamin A gene, which that is transcribed to translate. It is basically autosomal genetic disorder due to the autosomal mutation with aging symptom, and HGPS is genetically dominant gene, and the histone mark H4K20 methyl 3 is involved and caused by de novo mutation that occur in a gene that encodes lamina A or lamin A, simply lamina. So let's understand about for the normal person, so how lamina will work. So in this way, the normal cell basically contain a lamin A gene, a lamina gene, which that transcribe, and this basically it will be transcription. We need to translate the protein. Protein contain a R S Y double L G, the different type of amino acid, that is 50 amino acid. So the R is for tyrosine, S for basically is the different type of amino acid. We will not understand. So in this way, here is the any amino acid in the X side, while the double A means the amino acid aliphatic, and the C for cysteine. So in this way, this is the protein sequence. Basically, it is not. It is pre-lamina A protein. So remember about that, which that contain 50 amino acid, which that with the sequence of RSYLLG. So this is the amino acid. So this is the pre-lamina A. And in this the farnesyl group will be at, is known as the farnesylation in the presence of the farnesyl transferase enzyme used with a C-terminal cleavage. And in this way, the C-terminal will cleavage will lead to C will be cysteine will be left behind, and the ZEMS T24 gene activation for metalloprotease enzyme for cleavage. So this cleavage for this protein will lead to farnesyl group add, is known as the farnesylation process. So this farnesylation process will lead to cleavage, and in this way, the cleavage basically will lead to amino acid aliphatic will be removed with the other amino acid, can be possible, and in this the methyl transferase enzyme will lead to use to add the methyl group with the cysteine, and in this way the methylation process will occur. This is known as the methylation process. And the further on, the pre-lamin A will convert into the mature lamin After the upstream cleavage due to the ZAMPT24 gene activation for produce more metalloprotease enzyme for the cleavage, and in this way the cleavage side will be LLG. So the SY will be left behind the amino acid. So the Y amino acid whenever will be left behind will create the sticky end. This sticky end will combine together in the site of the nuclear membrane to hold on and maintain the shape of the nucleus. So a sticky farnesyl is added to lamina A protein, which help it to reach the nuclear lamina A. In this way, it will lead to upstream cleavage, will lead to LLG side to cleavage, and again this sticky end formation will be occur. In this way, the farnesyl group removing process is very important. If it will be removed earlier, so it is also bad. If removed later, so also bad. So it should be removed during the adjust of this mature lamina A gene protein. Sorry, the for short for normal person, the lamina gene activate to synthesis this mature lamina protein, and in this way the RSY site. And in this way, basically, first of all, the farnesylation will lead to farnesyl group add, and the lamina protein will lead to mature, basically pre-lamina, lead to mature lamina whenever it will be removed the farnesyl group. And in this way, whenever it will be removed, so first it will be add in the nuclear site where the adjacent site for the maintenance of the skeleton of the nucleus will lead to maintain the genetic material inside of the nucleus, and the farnesyl is cut off. So in this is the cut off to lamina A incorporation, and that process we have discussed earlier. So HGPS problem will lead to lamina A mutation. Let's understand about that. So for the lamina A mutant, in this way the basically again translation to produce 50 amino acid deletion. So this 50 amino acid type will be deleted here, which that is RSYLLG. You can search to understand. So I will also give the link to understand, and also give the information in the link in the description below, in the description box. So here is the translation. Will lead to farnesylation. Again, farnesyl transferase enzyme used to add the farnesyl group, but the important thing is that methylation will also occur, will lead to activate this methyl, but the RSYLLG gene amino acid will not present. So in this way, whenever it will be not present, so how to second cleavage formation will possible? So the second cleavage formation will not possible. So in this way, here is you can see this is the mutant farnesylated pre-lamin A. Basically here you can see which that cysteine not add with the G site and LL site, like the amino acid, which that is 50 repeated amino acid. So in this the progerin is a protein will become, this is pre-lamin A, the mutant farnesylated progerin, which this is the progerin protein which that not remove the cleavage formation. The second cleavage will not occur, so the farnesyl group will not remove, and in this way the farnesyl group when not removed, the sticky end will not create. In this way the sticky end will not create, so the skeleton of the nucleus will not possible. In this way the damage and the disruption, here is the remove, but here is the basically not remove during the second cleavage. First cleavage it will be removed, but here is you can see the second cleavage it will not remove the farnesyl group. So in this way the basically the DNA instability will lead to further on the aging phenotypes. So the treatment can be lonafarinib and the orphan drug, which that is cardiovascular monitoring, also the acetyl salicylic acid and the growth hormone treatment for the basically it is also important for inhibitor. So in this way the important thing is that the farnesyl transferase enzyme inhibitor also people use, but the popular person, let's understand about that, the Sammy Basu, 28-year-old, and the Hayale O'Quins, which that she passed away in 17 years, Sam Byrne, and the Michael Vandert, which that also died due to the progeria, and they are the best researcher of the progeria disease. So the summarization is the progeria, also known as the HGPS, Hutchinson-Gilford progeria syndrome, extremely rare genetic disorder that cause children to age rapidly, and cause is the lamina protein, LMNA gene. The lamina gene produces the abnormal protein called the progerin. This protein disrupt the structure of the nucleus. So I hope you make sense about that. Basically, the second cleavage will not possible, while for normal, the RSY side, the 50 amino acid will be missed out in the lamina mutation will lead to cause HGPS, the progeria. So the cause is basically is the I hope you make sense about that. So this was the video. I hope so. See you in the next video for this, and bye. Allah Hafiz.